root rot diseases. Let's see what we have prepared for you today. Okay. Well, I guess you are familiar with a picture like this, right? Sometimes we have that problem, but always wonder what are the reasons. Today, what I decide to do is to present or to talk about the pathogens that cause root rot diseases. Some are directly related with the root system, but other ones are soil-borne that go through the root system to cause diseases in the plant. So let's go with the names of the main uh, uh, pathogens related in some way with root rot diseases. The main one, in the main picture, you know about this. You have heard about Phytophthora. So Phytophthora species, Phytophthora lives in the soil. It's a soil-borne pathogen. That means it's there all the time. And this pathogen infects a wide variety of ornamental crops, vegetables, and forest species. How to pronounce? It is Phytophthora. That is also means that it's a plant destroyer. And actually, plant pathology starts with because of this pathogen with the disease famine. I think I have that here. Yeah, it was one of the first of the species studied more than 150 years ago, and we still studying Phytophthora. We are knowing every time with different techniques, laboratory techniques, we are able to identify more species now. But this was the first disease that was studied that caused by Phytophthora infestans. Now, where these two groups, the main group that, the group that Phytophthora belongs is called oomycetes. What is oomycetes? It's organism that is not really a fungus that is related with that in the way that it grows, looks like produce mycelium, produces structures similar to fungi, and that is the reason sometimes we say it's a fungi, but it's not. It is a group really called oomycetes. We're gonna review little things now. They're really closely related with aquatic organisms. And in the evolution system, there is known that it's a specific group that is called Ornamentals, but that is not the main thing here, but it's just for you to know that the reason that this is a different group. So from these oomycetes, there are more than 1,000 species of plant pathogens, like you can see here. So in the evolution, some of the species that are with a leaf, they attack plants. And in this list of these 1,000 species of oomycetes, at least 90 of them are Phytophthora and pathogenic for plants. Now, what is the main difference between fungi and oomycete? One of the easy ways that we can recognize is when we have a slide under the microscope. The mycelium, or these hyphae that you can see here, they have a septa, a division, like a little wall in some places in the mycelium. But in the oomycetes, it's like, it looks like they have a mycelium too, but it doesn't have this, the presence of this. That is one of the easy ways to differentiate sometimes, or give us a confirmation sometimes that these are Phytophthora or oomycete species. Now, with Phytophthora, we have different things here. We have different structures, reproductive structures. The vegetative reproduction by sporangia. So we can see here in the graphic in the left. So these are little structures that will contain a lot of little particles called like the zoospores. These zoospores are inside of the sporangia, and zoospores are called because they are motile. They move in the water. So I don't know, we can put a little slide here to, can we? I don't want to try. You can share, Maria. I can stop Maya and you can share that video. All right, thank you.
Yes.
So here what we can see is the specific is a Phytophthora ramorum, how the spores are inside the sporangia. As soon they have the media or the conditions, they will leave and go to attack or infect the plant. So the number inside of the zoospores inside the sporangia, it's going to be related with the species. So the number of zoospores varies from maybe 50 to more than 100 zoospores. That depends on the species. And you can see how they start to live like it was an egg, right? With many babies there. All right. Okay, thank you. So let's go back to our presentation. So these are the structures that how these pathogens reproduce, but also we need to understand this, what is the reason they survive for a long time in the soil and what are a threatening pathogen every time we put a plant in the soil. Is because they are there, all right, but also they produce these survival structures that could be there for a long time. So there are, I'm going to be brief in that. I'm don't give too much detail, but we have two groups of these. Some are called chlamydospores and others that are oospores, because some of these Phytophthora species can reproduce sexually and they form oospores structures. So these are the ones that they survive for a long, long time in the soil. We don't want them, but they are there. We cannot do that. This is a brief kind of life cycle where we can see the structures and how they move around until the plants get, how the plants get it. Well, this is the life cycle of Phytophthora. So we're talking about the survival structures that we can find in the soil, and then these it will maybe germinate, but if not, we have the sporangia, vegetative reproduction, that is the main way that the Phytophthora infect plants. Well, the thing is, we have a diversity, and initially, well, we do cultures, we try to isolate them, and you can see like here a variety of shapes, right? The morphology of the sporangia is one of the characteristics to identify or give a name. But now we have, as I said, techniques, molecular techniques that help us to do this identification really fast. Now, there are several studies trying to, identify as much as possible how many of these are out there attacking our plants in the nursery environment. And every time they're doing different studies, they start to find several species that maybe in the past were not identified. But I'm just going to share a summary of some paper they did. I think it's here in Oregon. I'm pretty sure it was in Oregon. A study about the diversity of species of Oomycetes, Phytophthora pythium, from recycling ponds. What I hear in the graphic in the left, well, they collected water, they have different bathing techniques maybe to collect the Phytophthora, or they just take the water, and then they do, in this case, they did two different techniques to identify or isolate this Phytophthora pythium. What was the reason they were doing here in the water? Why the water? Because in most of the nurseries, we have recycling ponds, right? We reuse the water and irrigate our plants. So that is the fear that what is going to happen, we are going to put back the pathogens if some of the plants get infected, going back. So they did this study and they found that depending on the technique, they can isolate more pythium species or Phytophthora species. And if they do leaf dating, that means we put some leaves that are really from plants that are susceptible for Phytophthora, we were able to isolate from them. So from doing this technique, they were able to isolate 63% of the species they were Phytophthora. And then Pythium was low. And there's another species here that you are going to see, or maybe you have heard about, that is Phytopythium. Phytopythium is a new genus, but it was based on, as I said, the studies of taxonomy based on DNA, and they identify as a new genus that now is really common in nurseries and was before classified as a Pythium species. That's what I can tell you about that, since it's in the graphic. And so if they do a filtration technique, they were able to isolate more species of Pythium than Phytophthora. So like you can see, depending how the study or the research do the isolation, they can identify more or less species. Just sharing, you talking about the diversity, right? That there are other factors also that we need to consider when we do research. Let's go back to, let's continue with the Phytophthora thing. We want to go to the root rods, right? And I'm taking too much time about Phytophthora, but talking about the biology, but I think it's important. So Phytophthora, it is a pathogen that produces different kind of symptoms, from wilting to complete death of the plant. Also, there are some species that cause aerial lesions or in the stems, but we want to know how do they do the root rods. So depending on the species, that there are some ones that are going to be specialized just to cause root rods, and others that could cause root rods and also a foliar damage. But that depends of the area, the species, and the time. Sometimes is related with the time of the year, but that could be considered. As I said, they can cause damage in the roots, but also when they cause foliar damage, we can see symptoms like these, like we see necrotic tissues, that means dead tissue, and we see symptoms like leaf spots, spots that are growing usually from the edge to borders of the leaf, where the water stands, where the leaf wetness stay, how is favored for that. So we have water in the leaves that will favor the infection of some spores of Phytophthora. The damage caused by these species of Phytophthora could be huge. There are species like, well, I'm not talking about Phytophthora ramorum today, but it's has this pathogen has a really wide host range, and is mainly, at least I think, more than 120 species of ornamentals that are could be or are susceptible for ramorum. So they could cause really damage. So ecological niche, I will say that all these ramorum has caused a devastating problem in the forest that is coming from California and extending to now Oregon in some areas where there are some quarantine areas that trying to control this pathogen to extend to other areas. And Phytophthora cinnamomi, that usually is present in the soil, as I said, and cause damage in several crops around the, especially around here in Oregon, around the Willamette Valley, you can hear some problems related with Phytophthora and berries. And there are some other species that could be in water. But I want to say something. There are some species that of Phytophthora, they are not pathogenic. They are there, but they don't cause any damage to plants. All right. Going to our root rot symptoms, what we can see when the damage starts in the roots, but then it's going slowly growing to the rest of the plant. So if the roots are damaged, the plants cannot take water to the plant. So we will have symptoms like wilting, yellowing, and eventually in some moment, the plant is going to die, right? Symptoms sometimes could take some time to identify the problem. So that is the thing that we need to be scouting and try to look the solutions. But we need to check, as we mentioned in other previous classes, the areas where standing water is present, and also where you can see the drainage of the water is poor because of clay soils. But in the right, you can see a photo of a plant that is completely bad. Yeah, and I don't think that happens one day to another. It took some times. In the left, there is a plant, and boxwood plants also can be affected by Phytophthora, but there is a difference with other diseases. But when you see part of one branch of the plant, like it looks yellowing and wilting, that sometimes usually is Phytophthora. It's different like boxwood blight, where the leaves are falling. These leaves don't fall. So that is one way to differentiate that is Phytophthora or other disease or boxwood blight. All right, so here are two pictures that I had from the past, get from some publication, I guess. In the left, we have a rhododendron wilting, dying, right? And this was caused by Phytophthora root rot. And the right, Azalea is another good host for Phytophthora. And you can see here the stunting and some lack of, the plant is not producing more leaves, and it doesn't look healthy either. So this was also caused by Phytophthora root rot. Well, let's talk about in container production, what we can see there. Here, this is from a publication from Jerry Whelan in 2016 about showing the damage of Phytophthora root rot. In the left, we can see a healthy radical system from rhododendron, and in the right, different stages, we can say that, of root rot damage caused by Phytophthora. Well, there is this concept that Phytophthora activity, it is different. The species are less active or not, depending on temperature, relative humidity, and this there is this nice, I want to show you a chart where show different species where they have been found and are related with the time of the season of the year. Usually some of them germinate or infect plants during the wet season, and symptoms could be seen during the dry season. So that is what I think, analyzing this data and doing some research, we have this chart here that was published several years ago, but I think it's from Oregon, to one of the researchers in Oregon studying Phytophthora species, where they isolated some species during some times of the year. Everything in green are species that isolated from foliar disease, foliar damage, so you can see here. So these, for example, they are not seen or present during the hot months or warmer months of the year. And then we have Ramorum that eventually could be seen any time of the year and isolated from foliar damage. And then we have these two other species that could be foliar, could cause foliar or root rot damage, or crown disease damage, that is Citricola and Cactorum in this case. And these species looks like more active, but no, that we can see the symptoms and growing maybe actively during warmer months of the year. And then we have cinnamomum, that is one of the main identified that cause root rot and crown diseases in several crops. So now it is my nursery at risk for Phytophthora. That is something that you have to decide, especially if your production system, in your production system, you have to put the plants directly with the soil. So then maybe it's a major concern, right? Because if you put your plants in containers, so you are controlling the substrate in some way, but there are other factors to consider. But here, depending on the production system that you have, you need to evaluate what could be your risk about Phytophthora. So here, for example, we have an area of, I think these are rhododendrons planted and close to an area of standing water. You can see the plants close to the water on the border are small, standing, right? They are growing close to the water, and they have less field space, and they are more kind of in danger for that. We also could have a standing water in places, even we have a barrier here, like in the, I don't know, cover area, but still we have a standing water, and we have two plants that look sick, and we have this standing water, and this, spread to other plants that could be a risk. So in order to do that, we need to check areas of points in our nurseries when we have standing water, and we need to take some correction actions in some way. I would like you to think about it. In the first picture, for example, we have here Phyris that is highly susceptible to Phytophthora too and root rot. And here in this nursery, you can see how sanitation practices is really poor. This plant looks, I didn't know if it's Phytophthora or not, but looked like had Phytophthora. But all these leaves with damage here stay there, and they were close to another. So there was a group of plants infected. The other didn't look that bad, but they had to pay attention to that if they don't want to get spread the disease in all the other plants. Here, even we don't have plants here, we have a standing water, and we have equipment that is moving around. So that is the other way to move the pathogens, especially these ones that we know they love the water, like Phytophthora and Pythium. So doing some considerations about this, always some things to review and think about it. So that your nursery have areas where there is a constant standing water, you need to think that, you need to direct that, like checklist or not. Is your nursery built in a slope and you have some standing water all the time? What we can do? Do you have a good drainage throughout your areas of production? Do you check for drains or some areas that could be clogged the water and you haven't noticed and produce again standing water? We don't want that. Do we have a greenhouse? Do we have some damage in the roof, some leaking, some problems with the cooling system, drips? All of that could be accumulated, and in some moment in long term maybe could cause the spread of some of these pathogens. And some of the practices, sometimes in greenhouses, we see that we have the hose just standing in the floor is wet and dirty, but well, I have seen that many places now they hang the hose nice. And at the beginning, when I started visiting nurses long time ago, still seeing the hose just in the floor and things like that. But I think people is understanding better what to do. Whatever is doing in cleaning and sanitation, it will prevent not only these kind of pathogens, but most of the pathogens. Here's another graphic trying to, researchers are always trying to identify these pathogens. Since we work with water and we work with plants, right? We need both. But if we don't have a good irrigation or we have over irrigation, we are providing the conditions for these pathogens to be there. So this is from a study that they did an evaluation. I don't know exactly the number of nurseries they did the sampling, but they were trying to identify the frequency of Pythium and Phytophthora in different areas in a nursery, from propagation, container, and field. So what they found here is that there are more, they were able to isolate more of these species from the field. So directly from plants were planted directly in the field is what I understand. Less species from plants that were produced in containers, and a little more in the propagation area. Considering these, we have this graphic here that you see in this pie diagram. So it is a little small, but it's just the frequency of the species. So in the propagation area, what they found more is a species of, in blue, a species of Pythium. So we're going to talk about Pythium briefly. And then in container area, they found a kind of 50-50, like a Phytophthora species and Pythium species. And in the field, where they have a mix, right? And here I think it's Phytophthora, different Phytophthora species, and less species of Pythium. So this is kind of the frequency of the species found in every area of production in a nursery. Here, there is another graphic to show part of the same study, where we can see here by nursery. As I said, I think they sample like 16 nurseries or something like that. And then what they found here, that is another thing. In every nursery, you will have different species of Phytophthora. Usually, well, there could be many other studies, right, to compare the same species are in one place another, and you can find the connection. Oh, they've sold plants to these nurseries, so they have the same species. I don't know. But only to show you that the species could be different in one nursery to another. What we consider as a take out from this, to me, is that whenever you bring plants from other place, you need to be careful. Even, you need to quarantine them. You don't know what is coming with the plant, so it's better to keep them aside. That is kind of my take message from the diversity of a species that you can find from one place to another. And compare with here in the across the nurseries in the A and B, you can see there are more species of Pythium identified than Phytophthora. And in the Phytophthora group, the most number or the highest, I don't know, percent of isolates were Pluribera. So Pluribera is kind of the predominant pathogen in these nurseries that were surveyed. Okay, so let's go to the best management practices for this. So one of the things we always recommend, we never put our substrates directly with the native soil. We know now that these pathogens live in the soil always. So we don't want to do that. We need to place our substrate in a clean area if it's possible, something that we can even, it's concrete, a barrier, we need that. Whenever you see a disease plant, we need to remove them. We don't know what is the disease, but we don't want that it is a disease to spread, right? We need to control the areas where we have standing water. We need to control our practices of sanitation. Here, look, this is what I meant when I see the hose laying in the, can you see, right? In the ground. So that, and then you irrigate the plants. How much soil, some, you know, the size of the spores, they can be spread easily. Bacteria, fungi, and then let's try to use always clean pots. I understand we can recycle them, but we need to take them through a good process of cleaning and sanitation before we use. And the small ones, I wouldn't even spend time to clean them. I will, if possible, I think it's cheaper to use new ones for small plots or things like that. I think will be safe to use new containers. Now let's go to talk the other mycet. I did too much importance to the queen Phytophthora. Let's go to the king Pythium now. Well, I'm sure you have heard about the damping off. What it means, the damping off is just when the seed is germinating, the plants just fall down in the crown, and you can see that is rotten. So Pythium is usually found in soil, it's a soil-borne pathogen, and attack juvenile stages of seedlings, and sometimes could be damaged before during the emergence of the seedling. It's common in propagation areas. It's high or less common in where you have mature plants in containers or in the field system. You have containers in the field. It's hard to find them there, but it's still there is a possibility depending on the susceptibility of the crop that you have. It favors by high soil moisture, we know that, and the pH around or greater than 5.8. So as Phytophthora, Pythium also has some structures, of course. If Phytophthora had, and this is a relative of Phytophthora, they also have these long-term survival structures. And they could still in the soil, in the flats, in the pots that you have, and they can stay forever. So that's the reason I highlight the fact that we need to clean any remaining soil that could be in your containers. If you think to reuse them, please do clean them. And there is another good practice or something that you should pay attention in order to prevent the spread of these pathogens is, I have seen in some places like you have areas or tables or benches where you work that are good benches. Those have a lot of cracks and it's so hard to clean or sanitize. So one of the things to improve in your practices is look for work areas where you have surfaces where you can clean and disinfect correctly. As I said, good benches could be the reservoir, not only for Pythium or Phytophthora because it's perfect, but for other pathogens too. Okay, Pythium symptoms, as I already started to talk about that, could cause damage before germination or emergence. And you can see how they look like in the picture in the right. Seeds could look black or soft or shriveled or sickly, like they are sick definitely. They don't look healthy at all. And a high soil moisture temperatures between 64 to 85 Fahrenheit will favor this damping off that we call when breaks like this. This is a lavender seedling, where you can see how it was attacked by Pythium. Pre-emergent damping off could cause by Pythium. It could be sometimes even lower temperatures, but combined with some high moistures, and also since there is a, they are not competitive microorganisms, let's say that one, that will be very efficient infection by pythium, even in lower temperatures. You continue with this. This is this is a conifer. This is a Douglas fir seedlings. You can see different stages of infection caused by pythium. And that's, I have seen pythium more in conifer production, in production of seedlings of conifers. That's when I have seen more pythium. And you can see here the roots start to turn brown to black and finally die. Other not even grow, of course, because they didn't have roots to keep growing, but the ones that are able to grow a little bit, then they start to get rotting in the few roots they have. Here, there's some pictures that we can see and the left completely root rot. And this was a geranium plant infected with pythium actually. So that's what it looked. Even the disease start to extend up to the stem of the plant. Here, this is a plantation of some Douglas fir, I think, and you can see here two plants. Could be Phytophthora, could be pythium, was in the lowest part of a slope. There was standing water, but in this moment we don't have standing water here. But you can see some of these seedlings never get to keep growing because of some pathogen in the area. And so always checking or doing scouting is a good practice to see what is going on and maybe control on time. We have done several studies or trials, efficacy trials on fungicides for Phytophthora, for pythium. And we usually do this in the greenhouse, but I just want to show you how much damage pythium could produce when we inoculate. And we want to see the diseases. And also we want to see if the fungicides that we use work. So we usually have these. We select the ones that survive and have nice looking, like in the right, and then the ones that are really, really affected by the pathogen in the left. Well, I have talked about the two main oomycetes, and then let's go briefly about other pathogens that are also soil-borne and cause damage to plants. But the next ones that I'm going to talk, like Rhizoctonia, is kind of an opportunistic, let's say that way. If the plants are in a bad environment and they are not the other soil-borne pathogens like pythium or Phytophthora, they are the ones that will attack. Rhizoctonia is a fungi. It's not affected by pH and increase when we have too much nitrogen available in the soil. It grows really nice with high temperatures. But this specific pathogen could be or could survive in dry conditions too. So it starts with the wet conditions. We're able to infect the plant, but after that, if it is there, it will cause like dry lesions. So that will be the difference. So Rhizoctonia is not a pathogen that I have worked in the past. I really haven't worked with this pathogen, to be honest, but it's there. And it caused damage in several ornamental plants. And the most common cause for Rhizoctonia is azalea, begonia, ferns, hollies, impatiens, and so on. And also they can cause stem rot. So we can see something like that here, like a poicentia. Everybody's familiar with that plant. We start with brown roots, and the tips of the roots start to decay, and there are some cankers start to form at the base in plants that are wilted. So you can check that and you can see, look for cankers or black roots or rotten roots, the tips of the roots. So the symptoms are kind of, that's the point. The symptoms could be something like, oh, it's pition. Oh no, if you don't see cankers, maybe you don't see rotten roots, maybe it is pition, but remember about the conditions they need. Also remember that pition is more attacking soft tissues or plant, small seedlings, more than adult plants. So that is something that helps. But also, if we don't know exactly, since we're talking about the mycetes and a fungus, and maybe it will be good to send a sample. If you cannot differentiate the symptoms, it's good to verify, maybe sending the plant clinic to analyze the sample. So these rhizoctonia root rot symptoms, the characteristics is this, like in the left, black, mushy, decaying, stunted plants, all the aerial parts or the part above the roots, right, will be wilt and yellowing and maybe some stunted plants. Here an example on the right, the petunias. And here the cankers could, so like you see here is like a dry canker, right? And then here, sometimes these break easily, they crack, so the stem will fall. And that will be something to see. It's not mushy, it's not, you know, watery. So it is, I think is definitely will be a good chance to be rhizoctonia there. And also this rhizoctonia could cause, you know, it is in the soil, it is splashed to the plants with water, with rain, could attack the aerial parts of the plants, causing different kinds of leaf damage or leaf spots, brown spots. And especially this you can see, well, not only in the field, but also in greenhouse, where maybe too much irrigation. And one of the things to differentiate is the growth of some mycelium that, well, I think you will be kind of a challenge to see that. But since in the greenhouse, we use good substrates, right? We try to use the best quality substrates. How do they get it? So, and then is when you have to identify what is your critical control point to see where you can bring to the greenhouse infected material and start to spread under that conditions. Okay, here are the pictures just to summarize what I'm talking about, the cankers and how to differentiate between pition and rhizoctonia. This is a good example. In the left and the right, we have these lesions caused by rhizoctonia, and in the center, we have the symptom caused by pition. So, as I said, pition causes more watery sub-root rot than in the lower part of the stem than the other one, like rhizoctonia, that cause more dry cankers and develops along the stems in the plant. So let's go to the other opportunistic soil-borne, that is Fusarium. So we usually don't talk about these pathogens, but they are there and they are terrible. And to be honest, Fusarium root rots are common in some of the ornamental plants, but I think these are kind of more threatening in tropical areas when the temperatures are high most of the time. I have seen Fusarium problems more in places where the temperature is high most of the time. So the reason we have this mentioning this pathogen in this list is because they infect plants by the roots and then go through all the plant infecting. And just you can see here, this poor plant doesn't have any roots and start to rot in the next. So it's caused vascular damage. And consequently, if they don't have a good root system, they're going to have a wooden you know, water or way to grow. Um, so how it is, the presence of this is just the presence of the susceptible plant will produce some exudates that make attract the pathogen to infect the plant. As I said, high temperatures are favors this pathogen to survive, and pH usually could be up to 7, and it will be a good uh, environment for them. So when we fertilize, things always about nitrogen. So nitrogen could make the plants grower, but prone from other diseases. So, and also the remaining, the excess of nitrogen in the soil could help to the prevalence of some pathogens too. So, and here another more symptoms, wilt, and sometimes we see just one side of the plant with some damage. And here, that is a nice example, a picture of damage by Fusarium root rot. All right, so what will be the Fusarium Pythium damage in this case? Let's do that. Well, we always start with the soil, the quality of substrate that we have. It is good if you have to use soil, if you don't have substrate, maybe you need to do that. Let's look for some alternative, maybe steaming the soil to have disinfected material. Steam will kill most of the pathogens, and some, even some weeds, seeds from weeds. Recommended to not reuse pots. Even they look that are clean, they don't have much, but always clean them if you think, clean and sanitize if you want to use it. Improve drainage or any in any container production. Watch for irrigation, depending on the, check for the needs of the plants for irrigation. Don't mix them once they need more and less. So try to consider the irrigation practice, irrigation system, the way that put together the plants that need more or less water. And soil, in this case, the optimal pH for this one, I guess, is 5, according to the literature, between 5.2 to 5.7, and ensure that the fungus nets are keep under control, because they can spread these spores, sporangios, and they can spread that. So be careful with them to control any kind of insect in general. There is some reference about biological control of Fusarium, and I found this that I like it, is Trichoderma hertzianum controlling Fusarium virguli. This is on soybeans, but I like it, the pictures kind to show, to understand how this Trichoderma could control a Fusarium species. So here in the left, we have a picture of IFA, a hyphae of Trichoderma, TH, and the here this kind of spore is a conidia from Fusarium that is germinating and form these, start to grow these hyphaes, right? And this is what you can see under the light microscope, but in the left is using another technique with a more sophisticated microscope, electronic scanning electron microscope, that you can see how the trichoderma keeps kind of strangulating the Fusarium hyphaes. So this is kind of, I thought would be interesting to see, take a, how it's used in some places. It seems like it's effective using some trichoderma to control, in this case, Fusarium. Oh, this is the same picture, but I was excited. Maybe this is better. You can see, you can see better this picture here and what I already explained. And now we have the last one. I'm gonna ask Maria to do the last slides. What about Maria?
All right, thank you. Okay. All right, so we are going to finish up by talking about verticillium, which I call an honorary root disease. It's not technically a root rot, and it's a vascular wilt, but just like fusarium that we just learned about, it attacks the plants through the roots. So it's just something to keep in mind when you're thinking about healthy plants and healthy roots. So verticillium wilt is caused by a variety of different verticillium species, but the most common that you're going to run into is verticillium dahliae. And now this particular pathogen has an enormous host range. It has hundreds of hosts that spans at least 14 plant families. And it includes a whole bunch of shade trees, fruits, vegetables, but also ornamentals. And you can see the list there of just some of the ornamentals that it will attack. There we go. All right, so let's look at the life cycle here for a minute, because it's going to become important whenever we talk about management. Now, verticillium is a monocyclic pathogen, and that basically means that you have the initial inoculum, but you don't have any secondary inoculum that's going to cause multiple generations per year. So your primary inoculum is down here, and this is the mycosclerotia, and it will germinate once it receives signals, chemical signals from root exudates. And it's going to penetrate the root tip, and it's going to start growing throughout all the roots, and it's eventually going to make its way into the xylem. And through there, it's going to basically colonize the entire vascular system of the plant. And it's going to form conidia, and it's going to grow all throughout the stem and the leaves. And this is going to start causing symptoms. And we'll go into symptoms in a second. But it's just going to continue to grow and grow. And as the host dies, it's going to start to form those really, really hardy mycosclerotia. And that's how it survives until the next growing season. So what are some symptoms? So symptoms are going to include wilt, yellowing of leaves. You're going to see some stems and leaves turning red or purple, stunted growth, and necrosis. And especially here on the right within the vascular system. So if you chop the stem and you see all this death within the vascular system, that might hint that you have verticillium. Now, only a laboratory can determine for sure whether you have verticillium, because fusarium and other wilt pathogens will cause similar symptoms. So how do you manage verticillium wilt? It's tricky. There's no way to cure verticillium. So if you have verticillium, you can't cure a plant of verticillium. So you have to focus on prevention. So prevention is key. And you can start by sanitizing pruning tools and equipment, because remember, the pathogen grows throughout the plant. So if you cut something and you have the pathogen inside, and then you just move directly to a clean plant or a healthy plant, you're just moving that pathogen. Remove infected material that could hold those mycosclerotia. Control weeds that serve as a host or alternate host for the fungus. Remember, there's hundreds of hosts. So if you have weeds growing out throughout the nursery, and even this becomes a problem if you rotate your crops, if you have field production, there's a lot of different hosts that could cause the fungus to survive. Investigate resistant cultivars, and soil fumigation is an option to reduce inoculum if you have it, but it will not completely wipe out all the mycosclerotia. All right, and that's it on